So in this video we will be discuss on the topic enzymes. Now the term enzyme, it was coined by Frederick Wilhelm Carney in the year 1878, and he coined this term for some active principle in yeast, which catalyze the fermentation of sugars to alcohol. So this term, the term enzyme now is universally used for all biological catalysts. Thus, the enzyme, it may be defined as the catalyst or as the catalyst of the biological system or as a biocatalyst. So here we see the definition on enzymes. They can be defined as the soluble, colloidal, organic catalysts formed from the living cells that catalyze a specific biochemical reaction by lowering the activation energy, and in the process they remain unchanged. So when we study in detail, you will see that in an enzyme catalyzed reaction, where the reactants are converted into product, in the presence of enzyme, the activation energy is low, whereas this shows the activation energy. This graph here, it shows the activation energy in the absence of enzyme. Whereas with enzyme, so in the presence of enzyme, the activation energy is low. So these enzymes, they lower the activation energy. They catalyze the biochemical reaction by lowering the activation energy, and in the process they themselves, they remain unchanged. So these enzymes, the study of enzymes, it started in the late 1700 with the study of digestion of meat by the gastric juice, and this study, it continues with the conversion of starch into sugar. It continues with the study of conversion of starch into sugar by the saliva and also plant extracts. And in the middle 19th century, a scientist, Louis Pasteur, this scientist Louis Pasteur, he suggested that the conversion of sugar into ethanol by yeast, it was catalyzed by ferments. He suggested that the conversion of sugar into ethanol was catalyzed by ferments, and he believed that these ferments, they could be active only inside the yeast cell, only within the yeast cell. And it was in the year 1878 where Carney, he coined the term enzyme, which became more popular. Whereas in the year 1897, another another scientist, Edward Buchner, he demonstrated that this fermentation process, it could be possible. It could also be possible by using the yeast extracts, the yeast extracts. And that is why this showed that the fermenting principles or the enzymes, they could be active even outside the cell boundary. Even outside the cell boundary. Unlike what Louis Pasteur has suggested, that these enzymes, they could be active only within the yeast cell. But Edward Buchner, he demonstrated that the fermentation, it could be possible by using the yeast extracts only. That means the enzyme, they could be active even outside the cell boundary. So the another person, okay, James Sumner, okay, James Sumner, he crystallized enzyme urase in the year 1926, and he found that the crystals were only proteins. So he crystallized, the first enzyme to be crystallized is urase. Urase in the year 1926 by James Sumner, and he found that these crystals were only proteins. And also another enzyme, pepsin, okay, crystallized in the year 1930, which was also found to be protein. So these studies supported that all enzymes were proteins, as I had mentioned that earlier. Okay, initially it was proposed that all enzymes were proteins, okay, because of these studies. These studies, they supported that all enzymes were proteins, and later on it was only in the year 1980, okay, in the year 1980, where Tom Seg and also Sydney Altman, these two scientists, they independently showed the presence of a catalytic activity. Okay, they showed the presence of catalytic activity of certain RNAs. Okay, of certain RNAs, and these were named as ribozymes. So these ribozymes also, they has the catalytic property, and by the end of the 20th century, the catalytic role of some DNAs, okay, of some DNAs were also observed, and these enzymes were named as DNA enzymes. So thus we can see the two main types of enzymes could be recognized: the protein enzymes, okay, and the nucleic acid enzymes. The ribozymes and the DNA enzymes, they are the nucleic acid enzymes, and we have the protein enzymes. So with the exception of some catalytic nucleic acids, all other enzymes are proteins, and each enzyme, thus it has a very specific protein configuration, okay, which has marked positions for binding with the substrates so as to keep them in close proximity. So now we will take a look at the okay, one more thing here, the reactants of the enzyme catalyzed reaction. The reactants are referred to as the substrates, okay, and remember that each enzyme, it has a it is quite specific, okay, it is specific in character, acting only on a particular substrate so as to produce a particular product or products. Okay, so enzyme is specific in character, acting only on a particular substrate to form a particular product. So now we will take a look at the structure of enzymes. So with the exception of the nucleic acid enzymes such as the ribozyme and the DNA enzymes, we know that all other enzymes, they are proteinaceous in nature. Okay, they are proteinaceous in nature, and the enzymes, they are larger than their substrate. Okay, the enzymes, they are larger than their substrate, and also different enzymes, they differ in their sizes. Okay, so enzymes differ in their sizes, vary in their sizes, and their size, it usually ranges from 62 amino acid residue to an average of 2500 amino acid residue. And if you take a look at the structure, only a small section, okay, this whole enzyme, it's not that the whole enzyme it takes part in a chemical reaction, only a small section of the structure it involves in the catalysis. Okay, and that small portion is situated to the binding site. You can see here this substrate will bind to this site. Okay, so these small section which involve in the catalysis are situated next to the binding site, and both the catalytic site, okay, the catalytic site and the binding site, okay, binding site meaning the site which the enzyme will bind to the substrate, and so both these site, they constitute the active site. Okay, they constitute the enzyme active site. So here this is the enzyme active site where the substrate will bind. Okay, and also it is in this place, in this region, in the active site where the enzyme will catalyze the chemical reaction, that is the conversion of the substrate into product. So both these catalytic and binding site, they constitute the enzyme's active site. Now coming to the structure, so on the basis of a number of polypeptide chains, the enzymes, they are of two types. Okay, the monomeric enzymes, that is this monomeric enzymes, they consist of only one polypeptide chain. Okay, only one polypeptide chain or subunit. And examples of monomeric enzymes are ribonuclease, then we have lysozymes, hexokinase, etc. Okay, so these are the monomeric enzymes which constitute only of one polypeptide subunit, and these are functional. These enzymes, they are functional in their three-dimensional or in their tertiary structure. And we have the next category, that is the oligomeric enzymes. These enzymes, they consist of more than one polypeptide chain, and these enzymes, they are usually functional in their quaternary structure. Okay, in their quaternary structure. For example, the enzymes which fall under this category are aldolase, okay, which consists of 4 chains or 4 polypeptide chains termed as tetrameric. And also another important enzyme under this category is rubisco. Okay, rubisco of Calvin cycle. So this enzyme also, it consists of 24 chains, 24 polypeptide chains. And another enzyme is enolase. And this enzyme, it has 2 polypeptide chain, that is, it is a dimer, that is, it consists of 2 polypeptide subunits. So this is on the basis of the presence of on the basis of the number of polypeptide chain. Now on the basis of the chemical nature the enzymes are also of two types. We have the simple enzymes and we have the holo enzymes or conjugate enzymes. So these simple enzymes, they are wholly made up of proteins. okay, and wholly made up of proteins, and the enzymes under this category are urease, then we have lysozyme, pepsin, trypsin, etc. So these enzymes, they are wholly made up of proteins, and therefore they are referred to as the simple enzymes. Now let us take a look at the second type of enzymes, that is the holo enzyme. So holo enzymes or conjugate enzymes, they consist of a proteinaceous part. Okay, they consist of a proteinaceous part, that is a part which is made up of protein, which is referred to as the apo enzyme. Okay, and they also consist of a non-proteinaceous part. Here, a non-proteinaceous part, and that non-proteinaceous part is called a cofactor. So these two together, they form a complete enzyme referred to as the holo enzyme. Okay, referred to as the holo enzyme. So you can say that the holo enzyme, okay, which is active, it consists of the apo enzyme, which is a protein part, and it consists of a cofactor, okay, which is a non-protein part. So a holo enzyme, it consists of a protein part and termed as the apo enzyme, and a non-protein part termed as the cofactor. And here, coming to the apo enzyme, it constitutes the major portion of the enzyme. Okay, the apo enzyme, it constitutes the major portion of the enzyme, and different enzymes, they have different sequences of amino acids and variable lengths of protein molecules. So the specific sequence of amino acid, it provides specific nature of the enzymes, which is important for the biological activity. And proteins, they are colloidal in nature, and therefore they provide a very large surface area per unit of volume, which is the characteristic of the enzymes. And only few enzymes, they are made up of entirely of proteins, and those are termed as simple enzymes. Okay, so only enzymes are entirely proteins, such as proteolytic enzymes. So now coming to the cofactor or the non-protein parts, these usually are of three types. Okay, the cofactors are usually of three types, that is the coenzyme, the prosthetic group, and the inorganic cofactors.